Why your blood type is a 20-million-year-old war record. Scientists have catalogued 47 different blood group systems in the human body. 47. Not 4, not 8. 47 separate classification systems, layered on top of each other, all sitting on the surface of your red blood cells right now. That number should stop you. Evolution doesn't do decoration. It doesn't build 47 overlapping molecular systems for no reason. If they're still here, they're doing something. Something important enough that losing them was, over and over, more dangerous than keeping them. The answer isn't really about blood. It's about millions of years of invisible war. Parasites, plagues, migrations across frozen ground. One brutal interbreeding event between two different human species, and one specific parasite that may have quietly shaped the blood of almost everyone alive today. Start at the beginning. 20 million years ago. Homo, your genus, didn't exist yet. Modern humans are roughly 300,000 years old. 20 million years puts you before almost everything. Somewhere in that distant primate family tree, a common ancestor of humans, chimps, gorillas, bonobos, and orangutans was already carrying two of the blood type variants you recognize today. Type A, Type B. We know this from a landmark study led by researcher Laura Segurel and colleagues at the University of Chicago, comparing a specific stretch of DNA across humans and multiple other primate species. The result was unambiguous. A and B didn't evolve separately in each species after they split apart. They were inherited together from one shared ancestor and carried forward through the entire branching tree. Martin Olson, a transfusion specialist at Lund University, called the evidence convincing. A shared, very old capability, he said, maintained straight through the divergence of the species. Your blood type predates Homo erectus, predates Homo habilis. It existed before your lineage even split from chimpanzees, roughly 6 million years ago. It's been running inside primate bodies longer than most mammal species alive today have existed at all. If something survives that long, across that many species, it's earning its keep. So what exactly is a blood type at the actual molecular level? Not a different color, not a different texture. Your red blood cells carry tiny molecular flags on their surface, chains of sugar attached to protein called antigens. Type A cells fly the A flag. Type B cells fly the B flag. Type AB flies both. Type O flies neither. The O, incidentally, comes from the German word ohne, without. Type O isn't a third option. It's the absence of the other two. Here's why any of this matters beyond a hospital form. Your immune system is obsessed with flags. It keeps a strict internal registry, self and not self. Anything carrying an unrecognized flag gets attacked. Your own blood type flag gets registered as self from birth, tolerated completely. But your immune system also builds standing antibodies against whichever flags you don't carry. Type A blood means anti-B antibodies on permanent guard. Type B means anti-A. Type O carries both. Type AB carries neither, which is why AB can receive any blood type without rejection. None of that evolved for surgery. Transfusion wasn't medically real until the 1600s and wasn't reliably survivable until Karl Landsteiner mapped the whole system in 1900. Your immune system was built for something older, something far more dangerous: pathogens. Many parasites, bacteria, and viruses carry surface molecules that closely resemble the antigens on your own red blood cells. Molecular disguise. If a pathogen's disguise looks close enough to your own type A flag, your immune system, trained since before birth to leave A alone, hesitates. The pathogen gets a partial pass, but someone with type O blood never made that concession. Their anti-A antibodies are already loaded, already waiting. No hesitation. Immediate attack. Different pathogens mimic different types. Different people end up vulnerable to different threats, which means a population carrying a mix of blood types is much harder for any single pathogen to wipe out than a population where everyone matches. Genetic diversity functioning as population-level armor. The clearest proof of this is malaria, specifically Plasmodium falciparum, the deadliest malaria species, responsible for the bulk of the roughly 600,000 malaria deaths every year, even now. Falciparum has a vicious trick called rosetting. Infected red blood cells stick to healthy ones, clumping into clusters that can block the tiny blood vessels of the brain, causing cerebral malaria, coma, seizures, and death, even with treatment. The parasite builds these clusters using the A and B antigen structures as anchors. Bigger flags mean stickier, more effective rosettes. Type O cells don't carry those flags, nothing to anchor to. Rosettes built on type O cells stay smaller, weaker. They fall apart faster. A rigorous study of 567 children in Mali, an area where malaria never lets up, found type O blood in only 21% of severe malaria cases, compared to 44 to 45% among children with mild malaria or no malaria at all. That works out to a 66% reduction in the odds of severe disease, just from being type O. That's not a marginal statistic. That's the difference between a bloodline continuing and a bloodline ending, replayed across thousands of generations in malaria-heavy regions of sub-Saharan Africa, where falciparum has been killing people for at least 10,000 years. Type O didn't win because it was universally superior. It became dominant because the deadliest parasite in that specific environment happened to be worse at killing the people who had it. That's not a triumph. That's a coin landing on one side, over and over, until the lucky side became enormous. But here's where it gets more complicated. Malaria doesn't kill evenly everywhere. It thrives in the tropics, where the mosquito vector does well. Move into colder climates, drier ground, the mountains of Central Asia, or the forests of Northern Europe, and malaria pressure drops sharply. Different diseases take over instead. And as ancient humans spread out of Africa into radically different environments, their blood started answering to different threats. A study in Scientific Reports examined genetic data from 22 ancient Homo sapiens who lived between 46,000 and 16,500 years ago, alongside DNA from 14 Neanderthal specimens. It found the process wasn't passive. People didn't just carry their African blood types unchanged into new continents. They hit new pathogens, and whichever blood type handled those pathogens best got selected for, sometimes fast, reshaping the local mix. The researchers identified a striking pause point. Genetic and archaeological evidence points to a population of early Homo sapiens who, after leaving Africa, spent roughly 15,000 years on the Persian Plateau, the highland region between the Zagros Mountains and the surrounding land. 15,000 years. Long enough, in evolutionary terms, for a semi-isolated population to generate genuinely new mutations and for any new blood type variant to spread if it offered even a small edge against the local disease environment. Researchers called it a genetic incubator, a pause in the outward migration where random drift and local pressure had time to build new diversity before people pushed further into Eurasia. The same pattern repeated further along the route: East Asia, Northern Europe, South and Southeast Asia. Each new environment, new pathogens, new shifts in the local blood type mix. That's why the modern global map of blood types isn't random. It's a record. Type O dominates globally because malaria shaped early Africans first, and pressure continued in tropical Central and South America. Type A runs highest in Central and Northern Europe and parts of indigenous Australia, regions where something other than malaria was doing the selecting. Type B forms a distinct belt from northern India through Central Asia into northern China, tracing old migration and disease corridors. Type AB stays rare almost everywhere, with slightly elevated frequency in parts of Japan and Korea. Every cluster on that map is a disease fingerprint. And then there are the Neanderthals. They lived across Europe and Western Asia for hundreds of thousands of years, interbred with Homo sapiens at least once in documented genetic history, and vanished from the fossil record around 40,000 years ago, leaving behind bones, tools, and 1 to 4% of the DNA in every non-African person alive today. Researchers at Aix-Marseille University, publishing in PLOS1, sequenced blood group genetics across 3 Neanderthal genomes and 1 Denisovan genome, covering 7 different blood systems. In most respects, Neanderthals looked recognizably like us. Type O existed, so did A and B, but the Rh system told a darker story. Rh status depends on a protein called the Rhd antigen. Present, you're Rh positive. Absent, Rh negative. In modern pregnancy, mismatched Rh between mother and fetus can trigger a dangerous immune reaction. The mother's body attacking the baby's blood cells as foreign. The Neanderthal Rh variants were partial, missing specific molecular pieces that complete, standard versions carry. Among Neanderthals, mating with other Neanderthals carrying the same partial variant, that wasn't a problem. Compatible with compatible. Then modern humans arrived, interbred, and Neanderthal mothers may have started carrying hybrid fetuses inheriting complete Rh antigens from human fathers, antigens their own immune systems could read as foreign. Failed pregnancies, dead hybrid offspring, reproductive success, already strained by competition and possibly climate pressure, dropping further. Lead researcher Stefan Mazieres told Live Science that Neanderthals carried an Rh variant that's genuinely rare in modern humans. Combined with evidence of inbreeding in Neanderthal populations, the picture is one of low immune diversity, poorly positioned for a genetic collision with a more varied incoming species. Their blood type wasn't only different from yours, it may have been part of why interbreeding with modern humans cost them more than it cost us. An honest caveat before we finish, because this story doesn't stay locked in the ancient past. The same trade-offs are still running inside you right now. Type O protects against severe malaria, but leaves you more vulnerable to severe cholera. The cholera bacterium binds more aggressively in type O intestines, and a 1996 gastrointestinal outbreak in Scotland found 87.5% of the people who died from it were type O. Type A carries elevated risk for certain cardiovascular conditions and some cancers. Type B shows its own distinct pattern. Researchers are still actively tracking links between blood type and COVID-19 severity, clotting risk, gastric cancer. None of these associations are simple or fully mapped, but none of them look accidental either. The ancient pressure that built this system never actually turned off. It's still running. Every one of your roughly 25 trillion red blood cells is carrying a molecular record of your ancestors' worst enemies, shaped by parasites that killed people with different flags, by plagues that left behind only the people whose blood was the wrong target, by a 15,000-year pause on a highland plateau, by an interbreeding event between two species whose blood didn't quite match. Your blood type isn't random. It's not a technicality on a hospital form. You are not broken. You're carrying a 20-million-year survival record, compressed onto the surface of the most common cell in your body.